Thank you very much. Thanks, Kim, for the great introduction, and thanks, Ido and other organizers for the nice invite. I'm very happy to join you guys here today to talk about some work that we've recently been doing, led by a student, Duncan Forster, I think is in the audience, in really great collaboration with Bo Wang, who's also, I think, in the audience, and is at the Vector Institute in Toronto, so an expert in machine learning, and Charlie Boone, who's a yeast geneticist in Toronto, also at the Donnelly Center. So this is an interdisciplinary project between genomics lab, a computer science lab, and a computational biology lab, with Duncan leading the project. And so just before I start, I wanted to just mention that a brief background about my lab. So traditionally, we've been focused on systems biology, trying to understand the genotype to phenotype relationship through the lens of pathways and interaction networks. And we've worked, as Kim mentioned, on a number of different open source software projects with a large collaborative team, for instance, Cytoscape for network visualization, with Trey, Alex Pico, Scooter Morris, who I think is also in the audience, Chris Sander, and others. Gene Mania, which is a bit relevant to the talk today, gene function prediction with Quade Morris, and with Chris Sander and Emmett Demir developing Pathway Commons, which integrates pathway information from a wide variety of pathway databases. We have 2.4 million human interactions there for download and use. So today I'm going to talk about network-based gene function prediction. I hope that the new work that we'll describe in a moment will be valuable to the function COSI and this community. By way of background, network-based gene function prediction works on the principle that should be very familiar to everyone here, of guilt by association, where a gene is predicted to have the functions of its neighbors in a network. Traditionally, this problem has been addressed with supervised technology. So there's a number of resources like String and Gene Mania that take many different types of networks and globally weight them and organize them into one unified network that can be used for this guilt by association gene function prediction. More recently, some unsupervised methods have been developed, like Mashup, DeepNF, which we heard from Meet yesterday, and others, SNS that Bo worked on a number of years ago. And the idea here is that, so the idea with supervised network integration is that you globally weight networks trained on some benchmark or a user query. So you're using prior information to figure out how to score networks so you can integrate them. With the unsupervised approach, you don't have a training set of prior knowledge. So the idea that one idea that people have used is you try to preserve lower dimensional patterns like clusters or modules across the network. So when you're integrating, you just make sure that those patterns are conserved, and that helps you score the networks. There's different methods that people have used: matrix factorization, autoencoders, edge reinforcement, that have all been successful in this area. And there's a few assumptions I wanted to cover also in terms of background. So one assumption is that biological networks are complementary. We know there are many different network types: protein interaction, genetics, functional interaction, and sequence similarity relationships. And the idea is that we can integrate these to overcome bias and noise in individual networks and strengthen so that the integration is stronger than the individual networks. We also have an idea that some networks are more informative about gene function than others, and a general idea that if you integrate more networks with more nodes and more edges, that's better, again, for gene function prediction. So the project that we've been working on, that Duncan has spent quite a while recently working on, is called Biological Network Integration Using Convolutions, or Bionic. So at least we have a nice name. Hopefully I'll convince you that the method is also quite valuable. So this method is a deep multimodal autoencoder that's similar to DeepNF that was presented yesterday, but we've added an additional component of graph convolutional networks, and I'll explain what those are and why they are useful. one interesting feature of our work is that it supports partially overlapping data. So networks can cover non-overlapping gene sets. This is important because when you're integrating many networks, they don't all cover the same set of genes. And if you are limited to looking at the intersection of those networks, the more networks that you integrate, the smaller your intersection becomes, and it becomes weaker and weaker at sort of covering the genome. And we also show that this network integration method is more scalable than comparable methods. And of course, and importantly, it has high performance on benchmarks. So this is a quick summary of the method, and I'll show you more about that as we go forward. So for people that do not have a deep learning background, one of the useful technologies that has been developed in unsupervised learning, in this case, to answer the question, how to train the network without a benchmark. So for the supervised cases like string and Gmania, basically the networks are trained, the network integration is trained to score the network so it maximizes performance on predicting gene function using gene ontology. But when you are just given networks and you're not given a benchmark, how do you figure out how to weight these networks so that you can integrate them without comparing them to any known benchmark? So the autoencoder idea takes input, encodes it into a low-dimensional hidden layer, and then decodes it to output. In our case, the input is networks and the output is networks, and then the process, this encoding and decoding process is optimized so that the output reconstructs or matches the input. So this is a deep learning system. It runs iteratively to build this learn this low-dimensional hidden layer or embedding. And the idea is that somehow captures the essence of the input by forcing it to produce output that can match the input. And in our case, we expect that these are network topological features like modules and other patterns. Okay, so how do we get networks into the neural network? So traditionally, this has been done with a pre-processing step where you convert your networks into vectors, which are then more easy to use in deep learning. And a popular tool for this is node-to-vec. With Bionic, we've incorporated the network-to-vector mapping in the learning process. It's not exactly a network-to-vector mapping, but it takes the network directly as input using the idea of the development of graph convolutional networks. So the idea of a graph convolutional network is that you take a network here. We've added self-loops. This network can be represented as an adjacency matrix. The nodes are labeled with a one-hot encoding, so each node gets a different value in an adjacency matrix. And then the adjacency matrix and the node features are basically trained to, as part of your training system, however that's optimized, to develop some learned features that are, you can choose the dimension of these. In this case, they're trained to learn some lower dimensional embedding of the network. And then we can have one of these graph convolutional networks for each input network, and we can train the whole system together. So how does that work? This is the general overview of Bionic. So we have a graph convolutional network for every input network that we have. If we have 10 networks, we have 10 GCNs. There's a little bit more details involved in how we actually use GCNs that I can get to if people are interested. And then each GCN learns low dimensional features, and these are combined. The combination method considers a couple of different criteria. In the end, you get integrated features that are then decoded back to an adjacency matrix, which you can think of as a network, and we minimize the difference of the output and the input, like I explained for autoencoders. So one of the interesting things technically with Bionic is that it can handle partially overlapping networks. One of the methods that we compare to is called mashup. I'll have the reference in a future slide, but one of the things we noticed about some of the previous methods like mashup is that when you have two networks that don't overlap, in terms of the nodes or genes that they cover, the embeddings that are learned are not overlapping as well. And in this case, we get embeddings that are related to these different parts of this Venn diagram. With Bionic, we've been able to constrain the learning procedure so that it forces these embeddings together. So if there are missing nodes in a network, we pad that out, and there's a masking procedure that's included in the learning procedure that also includes some kind of stochastic feature that kind of forces the learning system to reconstruct networks well, even if there's missing information between the different networks. And so that allows us to integrate more networks without losing genes. Okay, so to evaluate Bionic, we started by integrating 3 big yeast networks from three different types. So we took a network from Costanzo et al. Genetic interaction map, published in 2016. This is converted using Pearson correlations. We look at the correlation of the gene interaction vectors. It covers thousands of nodes and many edges. We took a Coexpression network from this paper that knocked out many transcription factors and measured the gene expression results. And we have a correlation network based on gene expression profiles. And we have a protein interaction network from Nevin Krogan that covered also mapped many protein interactions and complexes in yeast. So we also compared this to 3 integration approaches: a naive union of networks, just combining all nodes and edges across all the networks; ISEL, which is a matrix factorization approach published in 2019; and mashup, a diffusion state approximation method that was published in 2016, that both do the same problem of unsupervised network integration. We had 3 performance evaluation criteria. I'll go through these one by one. So the first one is coannotation prediction. So once we learn a set of features for each gene, and I should have shown you the features as an embedding to visualize it using UMAP, each gene is associated with a vector. In our case, I think it's 256 dimensional vector, so 256 numbers for each gene that place it in functional context with all the other genes. And those features, we can compute the similarity of them, for instance, using cosine, and then we can check, depending on how similar, we choose for a threshold to identify similar genes. By these features, we can compare it to gene ontology, biological process, intact complexes, and KEGG pathways to see if the features that we identify with given similarity are co-annotated in these resources. And we do that for all the different terms, compute precision recall, and then we sum up everything or take an average precision to get a final number across the entire database of terms. And so we have our 3 benchmarks that I mentioned here, and we can see the average precision performance for the different methods that I mentioned. And the higher number here is, the higher the number here is, the better. The union, okay, so let's say ISEL is, the union is this light blue color, ISEL is a bit darker, mashup is darker and still, and Bionic is purple. So we show here that on all of these 3 benchmarks for these 3 networks, Bionic is beating the other methods. One interesting thing that we found is that the union is actually does surprisingly well on this coannotation prediction. So we can discuss that if you're interested later. We did have a second performance evaluation that was module detection. So we hierarchically clustered the gene features, and we globally optimized the hyperparameters for this hierarchical clustering on the benchmark. So we tried to find clusters that best match the benchmark as a whole. And then we compared the resulting modules with the benchmark using adjusted mutual information, which is similar to adjusted rank index. And again, for these 3 different benchmarks, we see that Bionic outperformed the other methods. So that was great to see. Finally, we predicted gene function prediction based on the features using a supervised machine learning, in this case, a logistic regression classifier. And we basically measured the accuracy across many gene terms. We used some GoSlim categories here for Go biological process and cellular component. And we tested, after doing cross-validation for training, we tested on 10% of the data set of the genes that were held out fully that were not included in developing the classifier. So again, Bionic does well. Actually, interestingly, in this one case, the union is doing very well, but overall, we think Bionic is globally doing quite well. Bionic performs well on diverse functional terms. So we looked at a sort of small set of major function terms that was developed for the Costanzo 2016 genetic interaction paper that I mentioned. And if we just look at the average precision score, the darker colors being better, we see that Bionic generally does better than the other techniques that we mentioned, maybe not perfectly in all cases, and it covers a lot of, it does well on many categories, although there is an interesting range. So some of these categories, Bionic and actually all the other methods do better on than others. Here's an example integration where we take the raw data from, where we can see the raw data from the protein interactions, the co-expression network, and the genetic interactions for the proteasome complex. And when we put those all together, combine them with Bionic, this is what we get. So it's definitely a better view of this complex, including the known substructure of this complex. Okay, that was, I just showed you results on integrating 3 networks of different types. We then checked if Bionic performs well on integrating a larger number of networks. So Duncan collected genetic interaction, co-expression, and protein interaction networks to total 68. This kind of shows how well Bionic is doing when you have lots of networks. So some of the other methods don't do well when you have lots of methods. In particular, mashup fails, and it can only run on up to 5 to 10 networks. The mashup authors proposed using a SVD approximation in this case, so we did that, but that method doesn't actually do that well in all cases. So it's not exactly a fair comparison. So in any case, Bionic is doing really well here, and scores outperforms in all of our 3 benchmarks the other methods, sometimes dramatically. We also looked at combining 37 human protein interaction networks and got similar results. Okay, so I mentioned that Bionic scales well. We did a couple of scaling tests. So in this case, we took Yeast co-expression networks and we integrated either 2, 3, 7, etc. networks, and we tested the precision for the our benchmark that focused on average precision, the Coannotation benchmark. And if we look at all the methods that we're comparing, including these 2 versions of mashup, we can see that mashup does actually very well when it has a few networks. It matches Bionic's performance in this particular test with co-expression networks. But at some point, we can't run mashup anymore, and Bionic is able to run, and it continues to grow in performance as we add more networks. We also tried a scaling test over genes or nodes. So an increasing number of nodes that we can provide, this is samples from 4 human protein interaction networks, showed that Bionic again scales well, and the performance increases the more information you give it, which is expected for a machine learning approach. So we wanted to check one assumption I mentioned in the beginning. So I think it's safe to say that in the field, people generally say that if you are integrating networks, the more networks you have, the better, although I think people know that this is not generally proven. But if we look at the co-expression networks, for instance, when we're integrating, we definitely see this trend. So the more networks we have, the better the performance of Bionic. And we can also see that with the human protein interaction networks that we integrated. But this, it turns out, is not always the case. So the first set of 3 networks that we integrated has these performance measures. And then when we integrated 68 networks, we can compute those performance measures again. And sometimes the performance measures are better or similar or better, but sometimes they're worse for when we integrate 68 networks. So for instance, I've circled them here in red. The 3 network case does better in Go biological process module definition than when we have 68 networks. And we see a few different examples of this. So I think this raises an interesting question for us. Anybody who's interested in network integration is how do you choose the networks to integrate? Ideally, the machine learning method would choose that for you. We just give it all the networks and it would figure out how to best integrate them. But clearly we've seen here that it can't do that perfectly. And so that's an area of future work that we're interested in just working on and discussing. We also have an assumption that some networks are more informative than others. We can look at each network that was integrated. These are all these networks that we integrated and the different types, genetic interactions, etc. And we can see that, interestingly, genetic interactions are the best at, especially this Costanzo, this very large Costanzo 2016 map, are the best at predicting Go biological process coannotation terms. Not surprisingly, protein interactions are the best at predicting protein complexes, although genetic interactions provide a lot of information there as well. And interestingly, Coexpression is very good at predicting KEGG pathways. So again, this sort of highlights the strengths and of different network types. And ideally, we'd like to use these strengths and not lose information when we're integrating. So it just gives us a little bit of additional insight into what information is being taken from where from the input data. There's also, we looked deeply into this method in various different ways, and we're actually still doing that a little bit, and to try to better understand how it's working. And one of the interesting things that we discovered when we looked at modules is that some modules don't integrate as well as others. Just like I mentioned, that some gene function terms don't integrate as well as others, some modules don't. And I think that, you can think of modules as a type of gene function. And let's look at one example here. So these scatter plots, one of these scatter plots shows scores, basically performance scores for individual modules. So each module, for instance, this is intact complexes, is a dot here. And if we just look at the modules coming from an individual network, like the Krogan protein interaction network, which is defined to identify protein complexes, many of which are in the intact benchmark, then some of the modules score very well against the benchmark. They're perfect matches, probably because of the circularity. But what we really wanted to know is sort of how the modules score when we look at other networks or when we integrate them. So so each of these scatter plots compares modules, how well modules score if we look for modules in one network versus another. We have genetic protein interaction and gene co-expression networks here, as well as the union, sort of simple integration method, and bionic integrating actually the 68 yeast networks here. So what we found is that in general, bionic, the modules that we find from the bionic features score better than any of the individual networks, which is what we expect because we found that the general performance is higher. But there are now modules that score better in the individual network. So somehow we're losing information about some modules when we integrate. And that's going to be an interesting thing to follow up on to try and improve that, because ideally we'd want every module to be perfectly represented in the learning process and the ultimate features. Okay, so that's the bionic method. One of the one of the lessons that we learned here is that we spent a lot of time, so hopefully that method is going to be very useful for people and we can contribute to the dialogue in this area. I look forward to discussing this with the DeepNF authors as well in the future, since that was presented yesterday. And but one of the lessons that we learned is that we spent a lot of time evaluating the methods, and Duncan had to spend a lot of effort developing the benchmarks that we developed and organizing them. So because we couldn't find a standardized set of functional evaluations in the community, and so we had to implement our own. Our methods are not optimized, so they take a long time to run, and we've only captured 3 of them. I'm sure people here can come up with many ideas for how to evaluate these methods. And so what we'd like to propose to this community and anyone interested is, can we make a single library for integrating all of our benchmarks? So if you have features or networks that you're working with and you want to just test how well they're capturing known gene function, can we just make a library that is carefully designed and fast and optimized that can run to answer that question? So if you're interested, please contact us on the ISCB Slack channel or by email. I would be happy to collaborate on this project. Okay, so the last couple of slides, minutes of my talk, I just wanted to mention a couple of other projects that we're working on that are a bit about the future. And one of them, it relates to small scale network experiments. All the networks that I mentioned that we integrate with Bionic are large scale network experiments. So where a single paper has published a large amount of information about molecular or genetic interactions for a set of genes, but the literature and databases is filled with additional information, much more information from small scale experiments. And so how do we integrate that all together? So obviously we could just create a network that includes all the small scale networks, but one of the challenges that we have is that a lot of the information from these small scale networks is present in the literature and not in databases. So I'll talk to you about a little bit about a project that we have to try and address that challenge. And finally, I'll mention something about single cell genomics, which we've been very much involved in the past few years. Okay, so I mentioned the general idea that we want to get more network information in a computable form to use for our network integration and gene function prediction efforts. So lots of databases hire curators to read the literature and enter data into databases, which is incredibly valuable. Projects like Wiki Pathways try to crowdsource this information from the community, and that also works to some degree. Chris, Sander, Emek Demir, and myself have for a long time been thinking about a project that we now call Biofactoid that tries to engage authors when they publish a paper in the curation process. So it's a crowdsourcing idea, but it addresses authors when they're publishing papers. So the idea is that once an author's paper is accepted, then they can go to the Biofactoid website and very easily enter information about the experiments they've done, their results. And we're trying to work with publishers to recommend that authors use this technology to capture their information when their paper is about molecular interactions or other types of interactions. We have a lot of ideas here about how to incorporate text mining and how the people that are interacting with these systems to make computable knowledge need to also get something back from these systems. So we need to also think about how the computable knowledge can be converted back to something that people can use. And gene function prediction is one idea, but there are many ideas for how to make computable knowledge useful for authors. And we're thinking about that two-way translation between humans and computers that will help increase the efficiency, we hope, of this type of system in use by authors. Instead of authors just throwing data and not seeing where it goes, this becomes ideally would be somehow part of the tools that they use on a day-to-day basis, like Google. The second future direction I wanted to mention is related to single-cell genomics. I've been involved in this for the past 4 or 5 years. Half of my lab now is focused on interpreting single-cell genomics data. For instance, we had published a map of 5 human liver samples to make a human liver map. And what we see on these maps, where every circle represents a cell, and they're clustered by gene expression profile similarity, is that there's a lot of different types of cells. And the big discovery here with single-cell genomics is there are lots and lots and lots of cells and lots of subtypes of cells. And these cells have different activation states. And so it sort of gets you to thinking, once we start measuring all this information about cells, from a systems biology perspective, we've spent a lot of time as a community thinking about systems of genes within the cell. But now that we have lots of information about cells, thousands or millions of cells, what can we do with these systems? How can we extend our systems view? So today we think about gene function in the context of a cell. We think about cell lines, the cancer cell biology, the cell atlas. But when we start thinking about many cells together, we can think about ecosystems or systems of cells, and how do cells work together? What patterns are implemented and what's the function of these patterns? So for instance, we might be able to find patterns at the gene expression at the multi-cell level that are not visible for an individual cell. Like for instance, we might see a gradient that is present. And here are 2 gradients. You can hopefully see my mouse pointer, that are known to exist in the liver. So a gradient is an example of a pattern that is only the gradient of gene expression across many cells is an example of a pattern that's only visible when you have many cells. So the idea that I wanted to discuss with the community here is, can we extend the idea of function to cells? So what's the function of cells and these patterns? We have all the tools that we need. We discuss these all the time. So the question is, what's the function of a cell and these patterns? With that, I'd like to end my talk and acknowledge Duncan, Bo, and Charlie have done a huge amount of work on the Bionic project, and many people in my lab, past and present, for being a great team. Thank you.
All right, thank you, Gary. That was a wonderful talk. There's been a lot of activity on the Q&A. Duncan's been helping answer questions as we go. So what I will do actually is, let's see, perhaps I will skip over some of the ones that Duncan's gotten to try to make sure we get answers to most of them. Let's see, so we've got a question here. Why did you automatically assume every protein has a self loop? Not every protein is autocatalytic or forms a homodimer.
So that in the graph convolutional network is just a technical point that's needed for that process. And we can talk more about it on Slack, but it's not, it's an interesting question to sort of see how those self loops affect the results, but every gene gets a self loop, and it's just a technical point.
Okay. We have, let's see, looks like Duncan's got into a lot of these. Here's one. Okay, some of the precision values along the y-axis in your performance graphs were low, like 0.05. Shouldn't this be acknowledged? What is the value of a method that has this low a precision?
So that's a great question. And I think I've heard that question come up in other network integration methods. So there's a couple of answers to that question. So one, network integration is a research project. We're trying to increase those numbers. As you can tell, various methods over time has continually increased those numbers, and we hope that trend goes up. So where will that end? That's the research question. Can we do better? A second point is that we're comparing with benchmarks. Benchmarks are incomplete, and actually our you could maybe think that an integrated network with 68 networks or even 100 networks might do better at identifying modules than what we know is present in the databases. We don't have a very good way of assessing that, obviously, but we could, a future direction, obviously, would be to take modules that we identify that are not part of the benchmarks and experimentally validate them. Because we're connected with Charlie Boone's lab, that's the kind of thing that he does on a regular basis, so we hope to be able to do that in the future. We haven't done that yet. I think those are the two main points that I could mention with that. But it is the sort of general question of how high can we go is a very interesting one. It probably also relates to some fundamental aspects of our field, like how we describe gene function and how we, Trey Eidecker is doing a lot of work recently thinking about hierarchical views of gene function that are automatically determined. A lot of what we look at is just sets. Maybe we need better ways of describing gene function, and that can help us as well.
Very cool. Okay, let's see. We've got another question. Have you checked if the similarity of networks to each other when they perform better versus worse?
That's a good question. We didn't do that specifically. We could definitely try that to see if there's a pattern that we can find there. I think that that's going to be the real follow-up that we're doing, is sort of figuring out why certain methods do well and certain methods do less well. There could be many reasons. It could have to do with the noise in the network. It could have to do with the topological structures. That could also be multi-scale. It could have to do with the overall similarities and gene function areas that are covered, and what the multi-scale topological patterns that are associated with those functions look like. So there are many interesting research questions for us all to think about.
Great. Let's see. Did you find any interpretability challenge in using machine learning?
So we don't, I can show you what these embeddings look like. So this is an example of some Yeast embedding. So this is what we get in that's learned in the autoencoder. And if you are looking at these things and visualizing them and spinning them around in 3D, you can actually rationalize a lot of this similarity. So this gene that I highlighted here is part of a little highly similar cluster of genes that are all part of the secretion system. So from that perspective, we can actually see what's going on inside the box, but we don't know exactly how it's learned to put things together. And so that's the challenging part.
Another question, I think this will be our last one. Which yeast networks did you use?
So we have a list, and I can share with you if you get in touch with me.
Okay.
It's basically a lot of yeast networks that we collect from the SPELL database for co-expression and BioGrid for protein interaction and genetic interactions. It's almost all of the large networks that we get from those sources.